Okay.
Bueno, la doctora Dawn Holmes es investigadora en Western New England University y en la Universidad de Massachusetts. Sus aportaciones pues, han sido principalmente en el área de ecología microbiana, para entender la transferencia de electrones en organismos electroactivos para la producción de electricidad, de energía y bioremediación en sistemas bioelectroquímicos. Pero lo más sorprendente es las interacciones que estos tienen entre diferentes especies, incluso entre diferentes dominios, y es de lo que nos va a hablar hoy. Cuenta con alrededor de 100 publicaciones en revistas muy prestigiosas, entre las que encontramos Nature, Science, ISME, y cuenta con miles de citas. Entonces, bueno, esta es la introducción y ahorita hablaré un poquito en inglés para presentarla y ya que empiece, ¿no, Carlos?
Sí, por favor, Katy, ya en un par de minutos quizá ya.
In 2 minutes, Don.
2 minutes, ¿eh?
Okay,
2 minutes.
okay.
Sí.
Yo creo que ya podemos empezar, ¿verdad, Carlos?
Por favor, Katy, por favor, gracias.
Ok.
Gracias.
Okay, and it's a great pleasure for me to introduce our speaker today, who is going to talk also about direct interspecies electron transfer between Geobacter and various electron-accepting partners. Dawn, thanks for accepting the invitation, and could you start and share your screen?
Oh, yeah.
Okay.
Is that good? You're good? Can you hear me okay?
Yeah.
Okay.
Yeah, that's okay. Okay.
Okay, so today, should I start? Yeah, start?
Yes, please.
Okay, so today I'm going to talk about some different mechanisms that are used to transfer electrons between two syntrophic partners. So the electron donating partner in the studies that I've done is Geobacter metallireducens, and it can directly donate electrons to a variety of electron-accepting partners. So we'll learn about those. So I didn't know how diverse, how many different backgrounds there would be and, who I was presenting to. So I'll just give a kind of quick recap of what methanogens are and where you find them. So methanogens, because a lot of the diet studies that I've done have used Geobacter as the electron donating partner, and then a methanogen as the electron accepting partner to make methane. So methanogens are found in the domain archaea, and they're the only known organisms that can convert carbon dioxide to methane. And they use simple compounds like hydrogen, acetate, methanol, or formate to do this. And you find methanogens in strictly anaerobic environments, places like anaerobic digesters, deep subsurface soils and sediments, or in the intestinal tracts of animals. So methanogens can't use complex organic compounds. They're limited to the more simple compounds like C1 compounds or some C2 or C3 compounds. And they're classified by the substrates that they use to carry out methanogenesis. So you have three main groups. You have the hydrogenotrophic methanogens, which use the C1, which use hydrogen, CO2, or formate for methanogenesis. You have the acetoclastic methanogens, which use acetate as a substrate for methanogenesis. And you have the methyllotrophic ones, and they're the ones that can use methanol or the methylamines. So in addition to different types of methanogens being able to utilize the different substrates, they use different pathways. They actually use different enzymatic pathways to utilize these different substrates for growth. So if you have a hydrogenotrophic methanogen, it's going to use the hydrogenotrophic pathway. It's also called the CO2 reduction pathway. So it has different enzymes involved in the process, which ultimately feed into the last steps of methanogenesis. If it's an acetoclastic methanogen, it's going to use a different set of enzymes, which then ultimately feed into the final steps of methanogenesis. And then a different set of enzymes are used for the methylotrophic pathway. And this is obviously not a very detailed diagram of the pathways. It's just the important enzymes involved. And all of these steps share this final step for methanogenesis. So the majority of characterized methanogens are hydrogenotrophs, and they utilize the CO2 reduction pathway. And the acetoclastic pathway acetoclastic methanogens account for the smallest proportion of known methanogens. So the methanogens can be divided phylogenetically up into two different classes. So you have and so they diverged evolutionarily in two different directions. So you have the class 2 the class 2 methanogens and the class 1 methanogens. So the only known genera that are capable of acetoclastic methanogenesis come from the order Methanosarcinales, okay? And then within the Methanosarcinales, just the two genera that can carry out acetoclastic methanogenesis are Methanothrix, they're highlighted in red, and Methanosarcina. So in most environments, like places like sedimentary environments, sewage, cow rumens, or anaerobic digesters, the methanogens are found at the bottom of the food chain, right? So you start out at the top with the complex organic matter, which then gets degraded into smaller and smaller bits. And then down at the bottom of the food chain, that's where you find the various types of methanogens, which use the very simple compounds in order to make or produce methane. So that's how many methanogens survive, is basically by using fermentation products that are produced by fermentative organisms in their environment. Another type of metabolism, though, that methanogens can carry out is they can form these syntrophic partnerships with organisms like other bacteria. And when they form these syntrophic partnerships, this allows metabolisms that normally wouldn't be thermodynamically favorable to become exergonic, so to become favorable. So, for example, the conversion of ethanol to acetate and hydrogen is an endergonic reaction, so it's not favorable at high hydrogen partial pressures. But when the methanogen uses up some of that hydrogen, it creates low hydrogen partial pressures, and that makes the reaction favorable. So that's really a big role of the methanogens in these syntrophic partnerships, is basically making metabolisms that wouldn't ordinarily work and become exergonic. So there's two general types of syntropic partnerships between microbes, or between a bacterium and something like and between a methanogen. In one case, it's called indirect interspecies electron transfer. So when this happens, you have a fermentative bacterium that's producing compounds like hydrogen and formates, which then diffuse away, or they diffuse out of the bacterium. And then the methanogen in the nearby environment can then use that hydrogen and formates to produce methane. So this works, and there's a lot of this type of metabolism happening in environments like anaerobic digesters. But with this type of syntrophy, one problem is that a lot of the hydrogen formate diffuses away, and it doesn't good. Then the hydrogenotrophic methanogen never gets to see it. So in this other type of syntrophic metabolism between bacteria and methanogens, this is called direct interspecies electron transfer, or DIET. And in this case, instead of the methanogen getting electrons from some sort of compound like hydrogen, formate, or acetate, it's actually receiving electrons directly from the donating partner, and it's using those electrons for CO2 reduction to methane. So one feature of the electron donating partner that's participating in DIET is that it needs to be able to export electrons out of itself. Okay, so has to be able to get the electrons out of its outer membrane in order for the methanogen to then accept it. Okay. So in this example right here, this is what the first paper that really showed DIET between Geobacter and a methanogen. The Geobacter is oxidizing ethanol and then transferring electrons directly to the methanothrix species, which then reduces CO2 to methane. So I just went over indirect interspecies electron transfer, like in this example right here. Disulfovibrio is generating hydrogen, which then diffuses over to methanospirillum to make methane. And then over here, we have Geobacter oxidizing ethanol and generating a little bit of acetate, but then also electrons that then get dumped over to Methanosarcina barkeri. Another type of interspecies electron transfer is when a shuttle is used. So, for example, quinone-mediated interspecies electron transfer, which is also called quiet. This involves electrons being dumped onto a shuttle like AQDS, which then can bring those electrons over to the electron acceptor and do the same type of thing as that you see up here to reduce the accepting partner's electron acceptor. So one thing about quiet is that it's only been observed with Geobacter, 2 Geobacter species. And that's because fumarate is the electron acceptor in this case. And so AQDS, electron shuttle from AQDS to fumarate is energetically favorable. But every time that we've tried to do quiet with a methanogen, it hasn't worked. And I think that's because, so this isn't really a possible mechanism so far I've found for electron transfer to methanogens. And then finally, and this one it works with methanogens, it's where you use a conductive material as the interface in between the 2 organisms. So Geobacter will oxidize the ethanol, transfer its electrons to the conductive material, which is something like carbon cloth or granulated activated charcoal. And then the methanogen can pick up those electrons to make methane. So in order to get diet to occur, you need to have the 2 cells be able to come into close contact with each other. They need to be able to form tight aggregates so that they can, obviously, if they're transferring electrons, they need to be able to touch membranes, okay? So, so aggregates need to be able to form. And another important trait is that the electron donating bacterium needs to be able to transport electrons out of the cell. So mostly all of the organisms that we've studied so far that are electron donating partners are also metal respiring bacteria or exoelectrogens. And this means that they can get electrons out of their membrane and transfer them onto insoluble metals like iron oxides. So another important feature when you set up a diet co-culture is to use an electron accepting partner that ideally can't use hydrogen for growth, okay? Because then you can rule hydrogen interspecies electron transfer, you can rule that out of the equation. Or you can use an organism that doesn't use hydrogen. You could use an acetoclastic methanogen that can't use hydrogen, even if it is being generated. So during diet, Geobacter metallireducens oxidizes ethanol, and it produces some acetate to either methanosarcina or methanothrix. And then they use those electrons to reduce CO2 to methane. So the methanosarcina and the methanothrix cannot oxidize ethanol by themselves, and Geobacter metallireducens can't reduce CO2 by studies of diet and methanogenesis using the CO2 reduction pathway. So both of acetoclastic methanogenesis. So some methanosarcina can do both. Can use both acetate and hydrogen. So when we just focused on methanothrix, we knew that the only time it would use this top pathway would be if it was carrying out diet. And what we found was that methanothrix was expressing CO2 reduction pathway genes at really high levels, even higher than it was expressing the acetoclastic pathway genes. So this indicated that diet was in fact occurring in the flooded rice paddy sediments. And these sediments were producing methane also. We did the same type of study when we looked at anaerobic digesters treating brewery waste, found similar results. CO2 reduction pathway genes were from methanothrix were significantly upregulated. And then in another study, we looked at a microbial fuel cell that was treating sewage waste and also was involved in biogas upgrading. And we found not only that it was enriched with an exoelectrogen, specifically azonexis, so not Geobacter in this case, but azonexis and also methanothrix. And then when we looked further at the transcriptome of methanothrix, we found that it was expressing CO2 reduction pathway genes much more highly than the control reactor. In addition, we also found a bunch of methanothrix on the surface of the cathode that weren't interacting with the azonexis that also appeared to be accepting electrons directly from the cathode and also using the CO2 reduction pathway. So far, we were able to show with these studies that methanosarcina and methanothrix use the CO2 reduction pathway to produce methane during diet, but we still had two other important questions. So first, we wanted to know, how are the electrons getting out of the exoelectrogen? How are they getting out of the electron donating partner, through the outer membrane? And then second, how are those electrons then being taken up by the methanogen or any other electron accepting partner? So in the organisms that we focused on, we found that there were two general types of electron accepting partners, those that had surface associated C-type cytochromes. So that would include Geobacter sulfur-reducens was one that we've done studies with, methanosarcina, acetovorans, and methanosarcina subterranea. And then we also have accepting partners that do not have C-type cytochromes. And those include methanosarcina barkeri, the methanothrix species, and also the methanobacterium species. So even went further, you can even further classify within the methanosarcina. You have methanosarcina that have surface cytochromes and those that do not. So type 1 methanosarcina are the ones in red. They're also phylogenetically distinct, we found. So the ones in red, they lack outer surface C-type cytochromes, and most of them are capable of hydrogenotrophic methanogenesis, and they have functional hydrogenase proteins. The ones in blue, which we call the type 2 methanosarcina, on the other hand, do have outer surface C-type cytochromes. They also have a sodium translocating pump called an RNF complex, and they do not have functional hydrogenases. So this is a further breakdown of the two different types of methanosarcina. The type 1 are in yellow, and the type 2 are in green. And these are just a few examples. There's a lot more in the paper we published, but you can see for the most part that the ones in yellow, they do not have an RNF complex. They do, however, have an ECH hydrogenase complex, which is really important for hydrogen metabolism by methanogens. By these methanogens, they do not have multi-heme C-type cytochromes. They all use hydrogen as a source of electrons. And intriguingly, they are mostly found in organic rich environments where there's, there's just bigger source of hydrogen in those environments. Places like an anaerobic digester or a rumen fluid place, that's where you tend to find the type 1 methanogens or methanosarcina. The type 2, on the other hand, they do not have hydrogenases and they cannot use hydrogen as a source of electrons. And they tend to be found in places that are not rich in organic compounds, places like subsurface aquifer sediments. So when we looked at electron transport out of Geobacter metallireducens to the different, so we examined 5 different electron-accepting partners, okay? We found that there were big differences in the transcriptomic patterns. So Geobacter metallireducens seems to use different routes for electron transfer when it's paired with a different partner. But we did see certain patterns. So with Geobacter sulfur-reducens, it had one type of pattern. Then the type 1 methanosarcina and the methanothrix, both of which do not have multi-heme, surface-associated multi-heme cytochromes. So their membranes are probably different, in redox potential. They clustered together. So Geobacter metallireducens kind of treated them the same way. And then the type 2 methanosarcina, which both have multi-heme C-type cytochromes, their transcriptomes were similar. So it was very interesting to see these differences in mechanisms that were being used by metallireducens to get electrons out of the cell, depending on who the partner was. So specifically, we saw some big differences in expression patterns for genes coding for C-type cytochrome proteins in metallireducens. So in general, the C-type cytochromes that were most important for with Methanosarcina acetovorans and Methanosarcina subterranea. These are both type 2 Methanosarcina, and the patterns were similar. And then Methanosarcina barkeri and Methanothrix thermoacetophila, those patterns were similar. And then Geobacter sulfuriducens had very different patterns of gene expression for the C-type cytochromes. So this is pretty busy, but I just wanted to point out that we made a lot of mutants in Geobacter metallireducens, that we knocked out various C-type cytochromes, and then looked at how it impacted co-culture growth. And so we did a whole bunch of growth curves. Okay. We also looked at how we took a strain of Geobacter metallireducens that had a non-conductive pili, and we wanted to see whether or not that impacted aggregate establishment for the co-cultures. So we did find that when Geobacter metallireducens did not have a conductive pili, it did not establish co-cultures with Methanosarcina acetovorans or with Geobacter sulfuriducens after 300 days. When I let it continue to grow another, I guess it was like 365 days, eventually they did become established, but it took a really, really long time. So this definitely showed that the conductive pili were important for establishment of both Geobacter sulfuriducens and Methanosarcina acetovorans co-cultures. But when I looked at co-cultures of with Methanosarcina barkeri, they were able to become established within about 70 days with the non-conductive strain. And you can really see the differences in the aggregates through these TEM images. Over here, you can see that Methanosarcina acetovorans and Geobacter metallireducens, they don't need to be in direct contact with each other, right? They can be loosely apart, and then there are pili and archaea that appear to be connecting the two cells together. Over here, though, whenever we looked at the barkeri and the Geobacter co-cultures, they were almost always in direct contact with each other and almost made like a Geobacter methanogen sandwich. Okay, so in this case, you definitely wouldn't need pili, for long, distance electron transfer. So we haven't done the co-culture with the non-conductive strain of the non-conductive pili strain of Geobacter metallireducens and Methanothrix yet, but we're working on it right now. But we did find with the RNA-seq analysis that pili were not differentially expressed when we looked at growth in the presence of acetate versus diet, but they were much more significantly expressed in co-cultures with acetovorans and also with Geobacter sulfuriducens. And if you look at these images, you can see, just like with the Methanosarcina barkeri and the metallireducens, this is, right here is a Geobacter cell on top of a sheath of a Methanothrix. So they form these long sheaths. And so they're in direct contact with each other. And I think in those instances, you don't need to have the pili. They don't play as important of a role. So looking at all of those gene deletion co-cultures, we were able to see that there were slightly different routes for electron transport out of Geobacter metallireducens to the different electronic partners. So when the partners had surface cytochromes, we found that they used pili, or they needed pili, and also they used a different type of cytochrome pouring complex. So Geobacter metallireducens has 3 different cytochrome pouring complexes, which are, you have a porin channel, okay, and you have a periplasmic cytochrome, and you have an outer surface, outer membrane cytochrome, okay? And they form this complex that serves as a conduit that moves electrons from the periplasm outside of the cell. So the conduit, the porin conduit that seemed to be most important based on both genetics and on transcriptomic studies for these groups of co-cultures was called PCCG. And it was composed of a periplasmic cytochrome that we called OMAG, a porin complex that we called OMBG, and an outer membrane cytochrome that we called OMCG. So this was the porin complex when either sulfur-reducens or the type 2 methanosarsina were the accepting partners. This other outer surface cytochrome, 0930, which shares some homology to OMCZ and Geobacter sulfur-reducens, was critical for everybody. Okay, so this appeared to be a really important outer cytochrome, and it shares about 42% homology to OMCZ, and it has some other similarities. And with these guys, it's proposed that it then can transfer electrons to a conductive pili, or it can also probably transfer them directly to the surface of the cell. In the electron transfer to acceptors that do not have the surface cytochromes, it does not appear that pili are important. And another intriguing thing is that a different porin complex was important for electron transport out of the cell. This one we called OMAF, was the periplasmic cytochrome. OMBF is the porin. And then OMCF is the outer membrane cytochrome. And then it appears that those electrons then get passed on to 0930, which then transfers those electrons to the accepting partner. So one important feature of Methanosarsina barkeri that makes it a pretty nice model to study when you're looking at electron uptake mechanisms is that it can use both hydrogen and acetate. So it has both the hydrogenotrophic metabolism and an acetotrophic metabolism. So that meant that we were able to look at both interspecies hydrogen transfer and direct interspecies hydrogen, direct diet. We were able to look at both hydrogen transfer and diet with the same accepting partner. So in order to study HIT, that's what we call this, H is for hydrogen, we use Pelobacter carbonolicus as the electron donating partner, and it ferments ethanol and produces hydrogen. So in this case, Methanosarcina could then use its hydrogenase proteins to then get those electrons and make methane. Over here, we use Geobacter metallireducens, which does not produce hydrogen, and we looked at diet by Methanosarcina barkeri. And so this allowed us to compare differences in barkeri's metabolism using the two different types of syntrophic metabolisms. So we found that genes coding for hydrogenase proteins were highly expressed in both cells grown by diet and cells grown by HIT, but they were much more highly expressed when they were grown by hydrogen transfer. So the reason why barkeri still is expressing its hydrogenase genes, even though it's growing by diet, is because we found that even during diet, it uses hydrogen, intracellular hydrogen cycling. And this was the same as studies that showed that barkeri uses hydrogen cycling even when it grows on acetate. So this was consistent with the acetoclastic studies. But we also found that genes coding for subunits from the FPO dehydrogenase complex were much more highly expressed by barkeri cells grown by diet compared to growth by HIT. Normally, the FPO dehydrogenase complex pumps protons out of the cell to generate a proton gradient to then make ATP, right? And it gets electrons from reduced coenzyme F420. But it was proposed, and then also passes those electrons to methanophenazine. But it was proposed that it could operate in a paper. It was proposed that it could operate in the opposite direction, where something gives electrons to methanophenazine, which then passes them on to FPO. And then F420 gets reduced and the protons get pumped into the cell instead of out. So basically it can run in reverse. So in order for FPO dehydrogenase to run in reverse to drive methanogenesis, though, it needs to get its electrons from somewhere. So we looked at, with transcriptomic data, we looked at all of the surface and membrane-associated redox proteins that we could find to see which ones were more highly expressed in Methanosarcina barkeri. And we found 9 of them that were highly expressed. We just haven't yet been able to pinpoint which one is involved, but most likely one of these is involved in accepting electrons from the electron donating partner. So this was the mechanism that we proposed for electron uptake by barkeri from Geobacter metallireducens. So there's some sort of electron carrier on the surface. We don't know what yet, but it accepts electrons and then transfers those electrons to the lipid-soluble electron carrier, methanophenazine, transfers those electrons to FPO dehydrogenase, and then onto different components of the CO2 reduction pathway to make methane. So analysis of the RNA-seq data from co-cultures with Geobacter metallireducens and Methanothrix thermoacidophila showed similar results to Methanosarcina barkeri. So there's a lot fewer redox, surface-associated redox proteins in Methanothrix. So the only one that I could find that was upregulated in diet-grown cells was a quinoprotein. We'll have to, do more research to show, but right now, based on the data that we have, it looks like there is this quinoprotein that is exposed on the surface of the cell that could potentially accept electrons. And then the FPO dehydrogenase complex was also more highly expressed in the diet-grown cells. And so it could use a similar mechanism to Methanosarcina barkeri for electron uptake. Another interesting paper that came out recently showed that Methanothrix, it has a Rubisco shunt, and it can fix carbon, or it can fix CO2 into formaldehyde. And then that formaldehyde can feed into the methanogenesis pathway. And so just out of curiosity, I looked at these genes in the transcriptome, and I did find that the Rubisco shunt pathway genes were all being highly expressed. They were all well above the log 2 RPKM, the median log 2 RPKM values. And so they were being highly expressed, but they were also being highly expressed in the acetate grown cells. But it does look like they are using this pathway for growth, whether it's unique to diet or, just always. I don't know. Further research is needed. So finally, this was the study of the type 2 Methanosarcina, specifically Methanosarcina acetovorans. And these species, as I mentioned, have an RNF complex. And this is a sodium translocating electron transport complex. And it's unique to type 2 Methanosarcina. So it pumps sodium ions out of the cell when it's grown acetate. But it's also been proposed that it can work in reverse, just like the FPO dehydrogenase. And when cells were grown by diet versus when they were grown with just acetate, we found that most of the subunits from the RNF complex were more highly expressed when grown by diet than when they were grown with acetate. We also found several membrane-associated C-type cytochromes that were more highly expressed in diet-grown cells. And we found similar results when we looked at the transcriptome of the other type 2 methanogen, Methanosarcina. The RNF complex was up, and so were the C-type cytochrome genes. So we made some mutants in Methanosarcina acetovorans where we knocked out the gene coding for one of the C-type membrane-associated C-type cytochromes, MMCA. Actually, we knocked out all of the C-type cytochromes, but this one was the only one that had a phenotype. So when we removed this C-type cytochrome from our acetovoran cells, it was unable to establish diet. We also found that when we removed the genes for an Archaea, they also could not form aggregates. And when we examined the Archaea with more detail, we were able to see that they had the type 2 Methanosarcina archaea had many characteristics of pili, conductive pili, and also conductive archaea. So this species, Methanospirillum hungatai, it was shown to be conductive. And two characteristics of a conductive pilin or a conductive archaeolum are you need to have an abundance of aromatic amino acids, so at least 9%, okay? And the other one is you can't have a gap between the amino acids that's greater than 40. So we found there were a couple, so acetovorans has 3 different genes for archaea, and 2 of them appeared to be potentially conductive. But when we looked at the archaeolum gene from Barkeri, this was not the case. It did not have a lot of aromatics, and there was a large gap in between the aromatics. So the proposed mechanism for electron uptake by type 2 Methanosarcina is that electrons are accepted by MMCA, which is exposed on the surface of the membrane, and then they're transported either directly to the RNF complex or shuttled through methanophenazine. And the FPO dehydrogenase complex is also important, we found in for the type 2 Methanosarcina. And so this is what is proposed, different mechanisms for the different types of organisms for electron uptake. So there's been a lot of talk about how riboflavin additions help promote diet. And there was a paper that came out that showed that when riboflavin is added to co-cultures of Geobacter, metallireducens, and sulfur-reducens, that it enhances diet. And that's because fumarate is the electron acceptor in these systems. So transfer from riboflavin to fumarate is energetically favorable. But if you just look at the thermodynamics of riboflavin B, just like what I talked about AQDS, if you look at the thermodynamics, riboflavin transfer to CO2 is not favorable. And so when we did experiments, we found that the addition of riboflavin did not enhance diet between a Geobacter and a methanogen. So in conclusion, first of all, we showed that both Methanosarcina and methanothrix use the CO2 reduction pathway during diet. Second, diet between exoelectrogens and methanothrix or Methanosarcina is occurring in both natural and artificial environments, places like flooded rice paddy sediments or anaerobic digesters. Next, we found that Geobacter metallireducens uses different routes for electron transport out of the cell when it's grown in the presence of different electron accepting partners. Next, we showed that the porin cytochrome complex, OMAF, OMBF, OMCF, is most important for electron transport to type 1 Methanosarcina and methanothrix species. Those are the ones without the surface cytochromes. And then the porin cytochrome complex, OMAG, OMBG, OMCG, is the one that's most important for transport to type 2 Methanosarcina and Geobacter sulfur-reducens, both of which contain cytochromes. The outer membrane 8-heme cytochrome 0930 appears to be the most important cytochrome involved in electron transport out of Geobacter metallireducens out to all of the partners. And then the FPO dehydrogenase complex and potentially quinoproteins are involved in electron uptake by methanosarcina barkeri and methanothrix thermoacetophila. The outer surface cytochrome MMCA and the RNF complex are required for electron uptake by type 2 methanosarcina. And then finally, riboflavin does not enhance diet between Geobacter reducens and methanosarcina barkeri. Okay, and that's it.
Thanks, Dawn. It's a really great talk. And let's see if there is some questions.
Okay.
Okay. Mm-hmm. Okay, Daniel.
Thanks, Kathy. Dawn, thank you. It's a very nice work.
Oh, thank you.
I'm not an expert in this field, but I am used to thinking the indirect pathway for electron transfer between species. But this diet is for sure very important. It clearly shows a much closer cooperation between the different organisms than I previously thought. Your data shows that this direct interspecies electron transfers is very common. I like the example of the presence of CO2 reduction pathway in an organism that is not able to use CO2 directly. It's really nice. And the fact that the genes are upregulated during the interaction is really interesting.
Yeah, it makes it a lot easier to study than to study, because supposedly methanobacterium, I didn't do this work, but there's a methanobacterium, which is a hydrogenotrophic methanogen that supposedly can do diet, but I don't know how you would study that because it's a hydrogenotroph, right? So you're gonna be, I guess you could look at the hydrogenase proteins maybe and look at differential expression of those. But that makes it tricky, right? Because it only can use those genes.
Yeah. I wonder if you have some measurement of how much indirect versus direct electron transfer is happening in different environments. I think it's happening a lot, but have you measured that? No. How is the contribution of one or the other in different environments?
We wanted to. We actually wrote a grant. It didn't get funded, but that was our plan. Yes, but I agree. I feel like it would be very valuable to know, because it was assumed in the past that it was mostly indirect.
Yeah. Okay. Well, thank you. Very nice work. Thank you for sharing it.
Okay, let's see. I think it's no more questions. And I only want to mention that in the natural environment, there are many, many mechanisms that is even don't know for us. There are many, and I think it's very interesting that in two domains of the life, when you can think that there's no relation, they connect in the social phase to donate and accept electrons, and it is the fuel that moves all the metabolism in the anaerobic environments. And I think it's amazing, the mechanisms that even the mechanism that is not now, and maybe in many years you can see that there is for example, all the source of phase is cable and
I know. Oh, I know. Definitely.
it's very interesting. And let's see. Okay. And thanks. If there is no more questions, let's see. Okay. Thanks very much.
Yeah. Thank you
for sharing.
Bye.
Okay. Bye.
So bye.
Thank you very much. Gracias, Katy. Muchas gracias, Katy. Y bueno, nos esperamos el siguiente seminario. Muy amables.